Namaskar dear students, welcome you to our channel Gulab Sir. As a part of series of lecture, today's topic is types of enzymes. We learn about enzyme with their structure and function. Now, how many types of enzymes, major groups of enzymes are there which catalyze all the biochemical reactions occurring in living being? There are 6 major types of enzymes and are Oxidoreductases, Hydrolases, Transferase, Isomerases, Ligases and Lyases. These are the 6 major groups of enzymes that they control whole living world or all metabolic reactions occurring in all the living world. So it's very important to note about all these enzymes which catalyze all the biochemical reactions which occur in living world. So this is a group. There are, under that heading, there are many enzymes there, hundreds of enzymes that they work to have different kinds of oxidation and reduction reactions. As I told you, enzymes are specific in their action. It means that as type of reactions are there, that many types of enzymes are there, that many specific enzymes are there to catalyze that process. Therefore, although that major heading as a major part or major group, oxidoreductase is only one, but there are hundreds of enzymes under that heading which work for oxidation and reduction reactions occurring at various level, at various tissue or various processes. So here, suppose an enzyme NADH2 dehydrogenase is given under the heading for oxidoreductase and dehydrogenase. You know in chemistry what is oxidation? Donation of electron or addition of oxygen or release of hydrogen. These are the 3. So here itself 3 different kinds of process will be catalyzed by 3 different types of oxidoreductase enzymes, and thereby there is only one, that NADH2 dehydrogenase, which remove the hydrogen from NADH2 and add that hydrogen with PGAl to form 2 molecules of 3-PGA during the glycolysis process. Here that PGAl get converted into PGA. Thus, since the hydrogen is added into PGA, therefore it got reduced, and from NADH2 that hydrogen is released, therefore it is got oxidized. As a part of definition, you know what is oxidation reduction. So release of hydrogen is oxidation. That means NADH2 oxidized, therefore it is written there as oxidized. And together, that both the reaction, oxidation reduction reaction occur, therefore oxidoreductase. So this NADH2 dehydrogenase is oxidoreductase enzyme because it has caused both oxidation and reduction during that process. Oxidation of what? NADH2 and reduction of what? PGA. And therefore together this NADH2 dehydrogenase can be called as oxidoreductase enzyme. Now second type of or second group of enzyme, hydrolases. Lases matlab breakdown, hydro matlab addition of water. This group of enzyme which cause breakdown of complex organic compound into simple with the release of water. Therefore, it is called hydrolases. The process of breakdown of complex organic compound into simple with the loss of water is called hydrolases. And the enzyme which catalyzes this process is called as hydrolases. So you know that in digestive system, more than 10 to 15 enzymes that they play a role of breakdown of complex organic compounds such as protein, carbohydrate like starch and fat. For protein, you know that aminopeptidase, carboxypeptidase, trypsin, chymotrypsin, pepsin, these are the enzymes that they are hydrolytical enzymes. Amylases, salivary amylase, pancreatic amylase, intestinal amylase, they are also hydrolytic enzymes. Lipases, again intestinal lipases and pancreatic lipases, they are also hydrolytic enzymes because they break that complex organic substance into simple with the loss of water, and hence it is called as, see here this is process protein breakdown into amino acid with the loss of n number of H2, as many as number of amino acid in that chain will get break by different kinds of proteases. These enzymes as a group called proteases, that they break protein into amino acid. So depend upon the hundreds number of amino acid linked with that protein or linked in a chain by polypeptide bond to form protein. When they break, they produce hundreds of H2 because we have that very meaning of this enzyme is hydrolases, that with the loss of water. So you got it. that group of enzyme which cause breakdown of complex organic compound into simple with the loss of water, and hence hydrolases. Now here the transferase means something is transferred from one substrate to another substrate, and here it is shown substrate 1 which has that G as a group. That transfer with substrate 1, that is dash S dash, is this another substrate. So the group which was attached with substrate transferred to the substrate 1. That means that group G is transferred from substrate to substrate 1 during that process. That is called transferase or transformation. So here phosphate from adenosine triphosphate is transferred to the glucose in presence of phosphorylase or hexokinase enzyme during glycolysis, and hence glucose 6-phosphate is produced. The substrate glucose converted into glucose 6-phosphate. So required phosphate, it is transferred from ATP to the glucose in presence of enzyme phosphorylase. I hope you got it. That is what something transfer from one substrate to another substrate in presence of enzyme. That enzymes are called transferase. So examples of transferase enzyme: phosphorylase, hexokinase. These are the examples of enzyme transferase. Next, see the another type. See the another type, isomerase. You know that what is isomer. Isomers are nothing but which has same chemical formula but different structural formula. So during that process, that one substrate, that isomer, get converted into another type, that is another isomer. For example, glucose and fructose, they are isomer of each other because their chemical formula is same but structural formula is different. In that process, when glucose converted into fructose, that is why glucose 6-phosphate get converted to fructose 6-phosphate. 6-phosphate is common, glucose converted to fructose. What is fructose? Is the isomer of glucose. Only rearrangement of hydrogen and hydroxyl group occur, and therefore that reaction is called isomerization, and the enzyme which catalyzes is called isomerase. So hexokinase isomerase that convert glucose into fructose, and hence that enzyme isomerase is very simple. I hope again you got it. Ligases, that what we join, ligate, that what joining during DNA replication. Okazaki fragments are join each other in presence of ligase enzyme, and hence it is called that ligases which ligate Okazaki fragment. Another example, that bond between carbon oxygen, bond between carbon sulfur, bond between carbon and nitrogen, bond between phosphorus and oxygen, that what also catalyzed by that same enzyme ligases. Hence that the reaction which occur to join two substrates together, that enzyme commonly called as ligases. Another type of or group of enzyme, lysases. See, hydrolases and lysases are different. Hydrolases, those which catalyze the process with the removal of water, and lysases which catalyze the breakdown of complex into simple without loss of water, and that is the major difference that you have to remember. Here the example, lysases that cause the breakdown of bond between carbon and X and carbon and Y. So as without loss of water, to balance that valency of carbon, double bond is produced between these two subsequent carbon with the removal of X and Y without loss of water. So there are many substances in hydrocarbon chain, that suppose 3-chloro 4-bromo pentane. Chlorine and fluorine, suppose chlorine and bromine is removed. Naturally, double bond will produce between two subsequent carbon as to balance the valency of the carbon without loss of water. So 2-chloro and 3-bromo get converted into pentane, simple pentane, with the loss of chlorine and with the release of chlorine and bromine without loss of water, and that process is called lysases. So many examples, many reactions which where that some of the substrate get released from very common substrate without loss of water, that is the lysis, and the enzyme which catalyzes this process is called lysases. I hope you got these 6 major group of enzymes, how they work, that is written in your book. So question can be asked for 3 marks. Explain 6 different types of enzymes that catalyze the biochemical processes, begins with oxidoreductase. So please remember with suitable example. So once again, to sum up this video, the question which can be asked, write 6 different types of or different groups of enzyme with suitable example. I hope very simple video, you got it. Even if you are in doubt, you can ask in comment section. I appeal you to like and share our channel as well as Subscribe if you are new. Also, I request you to press bell icon to receive new notification as and when new videos are uploaded. Thank you. Enjoy the learning.